So we need to use a microscope in microbiology. It's one of our most common tools because we can't see any of these things with the naked eye, again, with the exception of some of our large multicellular worms. So microscopy is a really important skill for anyone to have that plans any sort of career in microbiology, and that would include things like clinical laboratory science. So any of you interested in like CLS, or you might be taking patient samples that you want to look at under a microscope, you really need to know how to use one, and to understand the principles of them. So that's what we're going to cover today. When we talk about size and what we're looking at under the microscope, we use the metric system, which is what the rest of the world uses for everything, but we only use it for science. So we use the metric system for science. The standard unit is a meter. If you don't like the metric system, that's okay. A meter is approximately-ish 3 feet. Okay. A little bit longer than 3 feet. The nice thing about the metric system is that everything is in units of 10. All right, how many feet are in a yard? Okay, 3. How many feet are in a mile? Like, that's really annoying. Okay, but how many meters are in a kilometer? Well, kilometer, 1,000. They make it really easy for us. So everything is in units of 10. When we talk about mass, like we talk about grams, kilograms, right? It's all that same system. So when we're looking at microscopy, we're talking about distance or length usually. So we talk in meters, but not in a microbe being a meter long. Again, that's multicellular worms, will be meters long. But really, an individual microorganism that you're looking at under the microscope will likely be on the micro or nanometer scale. So a micrometer right here is 1 millionth of a meter. A nanometer is 1 billionth of a meter. So that's the size range of a lot of these organisms, and we can't really discern that. So that's why we have to use the microscope. So, I'm not expecting that you memorize all of these conversions off the top of your head, but, theoretically, if you're being asked to multiply or divide by factors of 10, I would expect that you'd be able to do that without a calculator. And that will come into play in other, sections in the class as well. I'm not going to ask you to multiply 17 by 57, but, 10 times 10, 4 times 10, like those kinds of things you will have to do without a calculator. So it's just factors of 10. So 1 micrometer, right? 10 to the minus 3 millimeters, 10 to the minus 6 meters, a nanometer, right, just on that extra scale. 1,000 nanometers is 1 micrometer. 1 nanometer is 0.001 micrometers. So again, this is the scale we're talking about. If you picture a meter, again, as being just over 3 feet, cut that into a billion pieces, that's the scale where we're talking. So that's just why you can't see it. Unaided is what we say. So you can talk to your friends. If a microbe is 100 nanometers in length, how long is it in micrometers? What do we think?
0.1 micron.
0.1. 0.1 micrometers, or 1/10 of a micrometer. 1.1 micron. So again, this is the type of math that you may be expected to do. in regards to microscopy. Factors of 10. And again, I'm not going to be, trying to, let's put in decimeters and centimeters. I'm not going to do that. I didn't do that right now. I told you that they're in units of nanometers and micrometers, which are based off of the meter. So those are going to be the ones I expect you to know. Okay, so when we're talking about the sizes of different organisms, one of the things that people often say is that bacteria are always smaller than eukaryotes, and that's not true. Viruses are always smaller than bacteria. That's not true. Broadly, we can put them into size ranges, but there are a lot of exceptions when we talk about that. So if we're looking at this image here and we're going from left to right with the circles, left is the largest thing that we will look at in this set of images, and of course right is the smallest. So a tick, that's the actual size of a tick, very small, can be seen with the naked eye. And of course, that's blown up on the screen, so that's not the actual size of a tick. But the larval and nymphal stages of ticks can be things like poppy seed size or sesame seed size. Poppy seed, sesame seed, and then just a little bit bigger. So that's about the size of a tick. Red blood cells are some of the smallest cells that we have, because in mammals, our red blood cells don't have nuclei, so they're a little bit smaller. About 6 microns, 6 micrometers. But you do need a microscope to see them. And those of you going into medicine will often have to look at blood samples. You can tell a lot about the health of a patient by looking at their blood under the microscope. You can see pathogens in the blood. If you see bacteria in there or fungi, that's a problem. Parasites, those aren't supposed to be there. You can look at things like malaria that infect red blood cells. If there's something in the red blood cell, that's usually a problem. The number of white blood cells, the ratio, their morphology. Does your patient have an infection? Do they have leukemia? All sorts of information you can tell by looking at the blood. But because the cells are so small, you have to use a microscope. And of course, as you've experienced in the lab, those of you who are in the lab or have taken a lab like that, if you want to see something like E. coli under the microscope, right? You have to really get that highest magnification possible, that really fun oil immersion lens. We like that one, right? It's great. It's so easy to find everything. It's tricky, but you'll get better at it the more you practice. Then we start getting even smaller. So you have a bacteriophage. It's a virus that infects bacteria. Those really are outside of the range of a light microscope. So the tool that you have in the lab, the tool that you would commonly use in any sort of clinical setting, is a light microscope. And here's kind of the limit of what it can see. So things like viruses almost always are so small that the light microscope isn't powerful enough for us to visualize them. So in that case, we would turn to something like electron microscopy, which we'll talk about a little bit later in the slides. And there are newer types of microscopy that are very, very powerful and can allow you to see the DNA double helix, or even the molecules deposited on the surface of a cell, the atoms, the individual units. So there are some really powerful tools, but for most of our purposes, unless you're going into a research career, if you're going into any sort of clinical career, using a light microscope is going to be the tool that you need. And so the first microscope, this is, well, not the first one, this is von Leeuwenhoek's microscope, about the second one. Right? It's called simple. It has only one lens. So if you ever hear of something called a simple microscope, that means it has one lens. But that's not what we use. This would almost be like using a magnifying glass, but it's a better lens. So, right, if you have a magnifying glass, that's a single lens, and you can see things that make them bigger. But this is much more powerful than just a magnifying glass. Instead, what we use to get even better magnification so that we can see even smaller things is a compound microscope. So a compound microscope has a series of lenses. Typically, in a micro lab, you use a compound light microscope, which means it's a microscope that has multiple lenses. In our case, it'll have two. And we use light, visible light, in order to visualize our samples. So we use a compound light microscope, which will look something like this. Right? Every microscope is going to be a little bit different, but if you you look at the structure, they're all going to have the same pieces. They might just look a little bit different depending on the scope. So we can start at the bottom. You have the base. Everybody has a base, something so you can set it down. What can be very important and what you'll spend a lot of time with are the focusing knobs. Usually modern microscopes will put both coarse and fine on the same knob. Coarse is the bigger one, and that actually does larger scale movements of the stage up and down. Typically, you only want to use the coarse knob when you're first visualizing your sample. So you want to make sure that you are getting your sample in the right plane in three-dimensional space. Once you've got it there, you're like, I can see it, that's there. If you move up or down, you lose it. Then you move to the outer knob, which is smaller, the fine focus knob. And then that's where you can tweak very, very subtle movements of the stage up and down in space to get it in focus for your eyes. And I don't know if your instructors have told you yet. I imagine they have. When you do your midterm, right, you have to do a gram stain in the lab, and you have to find it under the microscope. So you do your stain, you find it under the microscope, and the only thing your instructor is willing to do is adjust the fine focus, because we all have slightly different eyes. They're not going to go through the whole cycle and look for it. But that's the goal of the fine focus, that somebody, in theory, once it's been focused, could come, adjust a little bit, and see it. The illuminator is your light source, and you'll usually have a knob, the rheostat, that allows you to control the amount of light. One of the things that often students I see kind of struggle with when you're first learning to use the microscope is you don't put enough light in. You don't let enough light in. You don't adjust the light source at all while you're moving into higher and higher focus. The more magnification you're trying to use, the more light you need. You don't crank it all the way up when you're on the lowest magnification, because that'll hurt your eyes. But as you increase magnification, in order to get the best resolution, which we'll talk about in a little bit, you have to increase the amount of visible light. So you should be adjusting the light every time you go up or down in magnification. The diaphragm can control the amount of light that enters the condenser. So you could, I guess in theory, adjust that to control the amount of light that's going into your stage. The condenser will focus the light on the specimen. Light it'll do whatever it wants. You turn on light, it's just gonna go scatter. So you wanna collect as much light as possible and focus it through your specimen, which will be your microscope slide. So you'll have your stage. Here's your microscope slide on your stage. Remember, the knobs control moving up and down. You'll have a little knob that comes off the side, somewhere over here, that controls it moving side to side and like back and forth. Then it encounters its first lens, the objective lens. So this is where you get your first round of magnification. Typically, you have a 10, a 40, and a 100x objective lens. So it will magnify the sample 10, 40, or 100 times. And the 100 is your oil immersion. The light passes through the arm, the arm's where you carry it, passes through the body tube into your second set of lenses, which are what you look through up here, the ocular lens. That generally adds another 10 times magnification. So in order to calculate your total magnification, right, you multiply the 10 that comes from the ocular, and then the 10, 40, or 100 that comes from your objective. So you end up with 100, 400, or 1,000 total magnification. There are some light microscopes I've seen that can do 2,000 times total magnification, but those are a little bit costlier, and I think it's debatable the quality of the image you get. But beyond that, you're really not going to be dealing with light microscopes anymore. That's kind of the upper limit of magnification that you can get with the light microscope. So again, you can follow the path of the light, and that's how microscopy works. So the really important terms that I know were emphasized in the lab and are going to be emphasized in the lecture, so hopefully hearing them at least twice is helpful to helping you remember them and understand them. The major limiting factor for why a light microscope really can max out at like 1,000 or 2,000 times total magnification is because of resolution. Resolution is the ability to see two objects as distinct. So if I'm looking at a field of cells, do I see big purple blob, or do I actually see little coccus, little coccus, little coccus, little coccus, little coccus, little coccus? If I see big blob of cells, I don't have the appropriate amount of resolution to tell that they're different. So I can improve resolution. I'll tell you the easiest way, right? Adding more light. The more light you have, especially those shorter wavelengths of light, those improve the resolution. So you want to let in as much light as possible. And that leads to the second thing you can do to improve your resolution is you add the immersion oil. And they're kind of a similar idea. You need to increase the amount of light that's passing through your specimen. The immersion oil captures that light and prevents it from scattering. So as the light comes up through the condenser tries to give you as much light as possible shining through your specimen. Once it gets through your specimen, it can bend and go away again. What the immersion oil does is help trap those light waves and funnel them into your oil immersion lens. That's why they have to be in contact. Oil immersion is not going to do you any good if there's a gap between the oil and the lens. The light will come out of the oil and disappear. It needs to be in contact. The lens needs to be touching the oil, which is on the slide. That's the other thing that I see, when you're first learning microscopy, that sometimes they're not touching. So it's not going to help. So that's how you can improve the resolution. And it works because of refraction, the bending of light. This is why we make biologists take physics. Light bends. So how do we capture as much as possible? Well, light bends in air, could refract and miss the lens, which is what you're seeing on the right-hand side of this image. So the condenser lens did as much as it could to shine all the light that it can through our specimen, and then bam, air happened, and we lose it. So the oil immersion helps to prevent as much refraction as possible to give us that better resolution, that better ability to distinguish those two objects as separate. So resolution and refraction are two really important properties when we're talking about microscopy. So that's why you have to use the oil immersion lens. Questions to this point? Cool. Okay, well, let's start talking then about different kinds of microscopy. So light microscopy, again, for your entire career, it is most likely that you will never use anything except a light microscope. It could be a modified light microscope, do a special kind of light microscopy, but it's light microscopy. Using visible light to observe your specimen. So these are some different kinds of microscopy that are based with light. They include just a compound light microscope, dark field, phase contrast, fluorescence, and confocal microscopy. Like I said, you may end up in maybe one of these more advanced ends of light microscopy, but almost certainly the majority of you will end up only in light microscopy. So very classically, and what you are going to be doing basically every time you're in the lab using your microscope is Brightfield light microscopy. The background of the slide is illuminated and your specimen is basically what kind of blocks the light. This allows you to see a sample that has been colored or stained. Bacteria are basically, they're colorless. That's why you have to do the staining procedure. You can't see them unless you do some type of stain on them. Even when you're wanting to do blood smears, especially if you're trying to look at different kinds of white blood cells, you have to do special staining procedures to see like the internal structures that make all of the different kinds of white blood cells different from one another. So you often will do different kinds of staining so that you can take advantage of this bright field. Dark field is a less common application of bright field microscopy. I know we have you play with both phase contrast and dark field in the lab. Bright field will kill any living sample. It focuses the light so much that it basically is cooking the little guys that are on the slide. So when you do things like the hay infusion, there's actually a lot of living organisms in there, and it's cool to see them move. So using something like dark field, or what we'll see on the next slide, phase contrast, doesn't cook them. It kind of reflects the light away. And so what we see is a dark background, and your sample almost looks illuminated. So this can be helpful definitely when you want to look at a living specimen. It can be helpful if you're looking at something that's unstained because it will just light up. And it can be useful if you're looking at things like spirochetes, really thin microorganisms. So when I was a doctoral student, that's how we counted spirochetes, is we counted them using dark field because you can't see them on bright field. And you can't stain them very well, they're just too thin. So it can allow for that. Then phase contrast, I think, is really cool. I never used it that much, mostly in the teaching labs. But so you have direct and diffracted rays. So this also allows you to look at living images. It just doesn't cook them. And so, you see it's a little bit more of a complex system of how the light travels through. But it's really useful when you're looking at some of your unicellular eukaryotes. You can start to see the internal structures. Bacteria, you're not going to see it. Even looking at bacteria with like phase contrast or dark field, you might be able to tell the shape. It's a spirochete, it's a coccus, it's a bacillus. You're not seeing any of the structures of it. They're just too small. But with some of your eukaryotes, like here's a paramecium or some sort of other ciliate, you can see some of the internal structures. So they're pretty cool. In clinical medicine, though, sometimes we want to adapt light microscopy to look for very specific things. And so you may end up doing something like fluorescence microscopy. So you use UV light to excite a fluorophore, which will then emit visible light that you can detect. This can be used for looking for pathogenic intracellular bacteria. It can look for viruses that are inside cells. You can look for certain markers of the immune system. So in clinical medicine, there definitely are applications for fluorescent microscopy. And then confocal microscopy is a lot like electron microscopy, a lot like fluorescent microscopy. It is fluorescent microscopy, but it's a more advanced application. So with confocal microscopy, it actually scans each plane, so it moves up just a little notch every time, and it will scan through the whole plane of your specimen, and then computationally put all of those images together, and then you can visualize your microbe or your sample, whatever it is you're looking at, in 3D. So you have more of a three-dimensional picture of your organism. And you can use green is really common. A lot of people use a green fluorophore. Red and green provide a nice contrast for one another. You can also use blue and yellow. So you can stack up these colors as well. And so here you see red and blue together, or red and green, where you can see some of the cytoskeletal components, and then inside you can see the nucleus. I don't imagine you'd use confocal a lot in a clinical setting, but it is just kind of one step up from standard fluorescent microscopy. Questions about light microscopy? Okay, cool. But now we want to see other stuff, or we want to see things better. So we move out of the light microscope into the electron microscope. Again, this is not going to be a terribly common tool in a clinical setting, though those of you who might end up going to medical school, if you're at a big name medical school, especially one that's associated with a research institution, so you're at UC Davis School of Medicine, and you've got UC Davis, which is world-class research, Stanford, those kinds of places, they will have electron microscopes. They can be used clinically, but again, it's not terribly common. I imagine Emmanuel doesn't have an electron microscope. And definitely your primary care physician doesn't have one. Instead of light, we move on to electrons in order to visualize the sample. Just like I told you with light, the shorter wavelengths of light, the more short wavelengths of light you have, the better resolution. Well, there's a limit to the wavelength of visible light. And when you reach that limit, right, then you can get into electrons, which have an even shorter wavelength, which provides a higher level of resolution. So now we can start to visualize things that are smaller than 0.2 micron. There are two major types of electron microscopy. The first is transmission electron microscopy. So that's kind of what it looks like. You have an electron gun which shoots electrons. You have lenses that help to focus it. Here's your specimen that can kind of scatter the electrons, and then they land on a plate. So when you're looking at a light microscope, you're directly looking at your sample. Or you might be looking at a computer screen that's showing your sample, but you're looking at your sample. When you're looking at preparing a sample and visualizing using electron microscopy, you're basically looking, they call it a micrograph. You're not looking at the sample itself, you're looking at the representation of the sample. Because here's where you're looking, down here at this photographic plate. Well, here's where your sample is. So you're not directly looking at the sample, you're looking at the representation of the sample. You can stain them with heavy metals or salts for better contrast. And so here what they're showing you, this is, you can use them for eukaryotes, but you can see all of the internal structures inside of the eukaryotic cell. So with transmission electron microscopy, you can get resolution down to things that are about 10 picometers. So remember, if a micrometer is a millionth of a meter, A nanometer is a billionth of a meter. A picometer is one whole level below that. So it's a big shift. And again, remember with light microscopy, let's be generous and say the limit with magnification is maybe 2,000 times. With transmission electron microscopy, we can do magnification from 10,000 to 10 million times. Which is why you can use it to visualize even internal components of a virus, for example. So there are some disadvantages to transmission electron microscopy in terms of like the image that it produces. One, it requires that you are able to do ultra thin sections of the specimen. They do a freeze fracture procedure to make thin sections. So that could theoretically cause damage. It doesn't produce a three-dimensional image. And because of the way that you have to make those ultra thin sections, you can get artifacts like shrinkage or distortion. So it may not be the most accurate or clear representation, but you get a solid idea. And we can contrast that with the other kind of electron microscopy, which is called scanning electron microscopy. This one is fun. I think the images it produces are really cool. I like doing scanning electron microscopy. You are using similar principles. You have a beam of electrons. They will hit the surface of your specimen and then be collected to create the image. This is great for looking at surface structures, right? They hit the surface. You can look at surface structures. You don't have to do any sectioning of your sample. So here's, again, like a ciliate, and you can see the individual cilia on the surface. It's really great detail. Now the resolution is different. We're at 10 nanometers versus the transmission was what's at 10 picometers. Magnification anywhere from 1,000 to 500,000. So here you're looking at like some of the fruiting body of a slime mold, and then here again is another bigger picture of one of those ciliates. Scanning electron micrographs are very, very cool. It's difficult to visualize viruses with a scanning electron microscope. Bacteria, easy, easy, easy. You can see the structure of a biofilm, for example. And I believe, if I'm not mistaken, if I did what I thought I intended to do, the course image for this class on Canvas is, this right here, is a biofilm of bacteria. And I grew those bacteria, and our electron microscopist visualized them for me. So you can see like damage to cells. I mean, it doesn't come out that color. He made it pretty for me as a going away present. But you get this really great ability to clearly see structure and what's happening to cells under various conditions. So why are these not used in a clinical setting? Theoretically, they're really not that difficult to use. A scientist or clinician could be trained to use an electron microscope. I've known undergraduates who knew how to use one. Not that complicated. The cheapest electron microscope I've ever heard of is $250,000. The nicer ones run closer to $1 million. And this was, before inflation. So they're just not affordable.
Back in high school, we had a scanning electron that was like $73,000 with Hitachi. Would like loan it to us?
They would loan it to you? Yeah.
To use it in Fluorescence.
Yeah, we had a tabletop one at Fresno State that was about $250. Yeah. So, but a really good one, yeah, they run you a lot. I think the one we had when I worked for the military was $1 million, and they kept it in the basement because if it moves, you're toast, right? It's like when you take a picture. You're like, oh, it looks beautiful, and then, I don't know, your hand went like this, and now your picture looks like garbage. So it was in the basement, which had less movement, and it was on like an air hockey table. So if there was any fluctuation in the ground, the air would keep it steady. So it was pretty wild. Very cool technology, though. And we do learn a lot, but they are going to be more common in a research setting. They can answer clinical questions, though. But in order to visualize viruses, you're usually talking about a transmission electron microscope. And of course, there are even newer advantages or newer technologies. For example, this is called scanning tunneling microscopy. I've never done this. I don't know anyone that has one of these. But it uses a tungsten probe. And it basically scans the surface. And the resolution, 1/100th of an atom. That's, to me, a scale that's not fathomable. I'm like, I don't understand what you mean. You can resolve 1/100th of an atom, but it can provide close range, three-dimensional images of molecules. For example, here they're showing you DNA that has been like kind of blown out of the cell. They color it. It's not obviously this color. So they're trying to indicate in yellow is going to be a chromosome, and then this purple one is probably an extra chromosomal piece of DNA, like a plasmid. And then this one I thought was really cool, atomic force microscopy. Again, you're getting that atom level detail. So it uses a metal and diamond probe to scan the surface, and it can produce three-dimensional images basically at that atomic level. So this is an image here of the pores formed by a Clostridium toxin in the surface of a cell. So each of those is a pore that's been formed in the surface of a cell by individual units of a Clostridium toxin. So it's pretty wild, like the amount of detail you can get with some of these types of microscopy. And again, you know, this is from the most recent edition of your textbook. Sometimes these are even not the newest types of technology. And I would say the cost goes down, but again, you know, inflation. And a lot of these are manufactured overseas, so there's tariffs and things like that. So now the cost has gone up again. But often what we see, even with really cool technologies like this, is the longer the technology has been around, the cheaper and cheaper and more accessible it starts to get. So hopefully we'll see some of those prices come back down and more places can get this type of detail from their samples. Okay, question about different kinds of microscopy. Okay. So how do we prepare samples for microscopy? And this is really going to focus on how we prepare samples for light microscopy. So again, those of you in the lab, you have hands-on experience doing this. Or if you haven't done staining yet, because I don't know what your actual schedule is, you will very, very soon. But we do it because live, unstained microbes aren't really able to be visualized just under Brightfield microscopy. They don't have enough contrast in order to see them under the microscope. So by staining, and depending on what type of stain you do, you can at least see the cell. Maybe if you do specialized staining for particular structures, you may be able to see those. So the first step that we do when we're preparing almost everything except when we do a negative stain is we make a smear. So you take microbes, you make a thin film on the slide. It's a little bit easier when you take them from liquid culture. You take a couple loops and spread it out on the slide. If it's from a plate, you put a little drop of water, mix a colony in there, let it dry. And then we fix them. You can fix a couple of different ways. You can fix using methanol. Methanol just produces wastes that we don't want to have to deal with. You're not really supposed to like pour it down the drain and things like that, especially in the quantities you would need to fix. So we use heat. That can have its downsides if you don't apply the heat enough, they won't be adhered to the slide. If you put too much heat, the cells will blow up. So, generally when you're doing heat fixation, it's kind of a, 1, 2, 3 gentle passes through the flame, and that's going to be sufficient for your microscopy. But you do want to let it air dry because, again, if it has that water in there, it can prevent them from adhering. And if you really hold it under there while it's got water, it's just going to boil them and make them explode. So we use dyes in order to visualize. So again, Staphylococcus isn't purple, it's just for that particular type of staining, it's a purple dye. I can use any kind of dye I want. I can make it blue, I can make it pink. Whatever kind of dye you use, that's what color the bacteria are going to be. So really, really commonly, you will use a basic dye. And the dye has, the chromophore is a cation. It means it's positively charged. It is attracted to the surface of the cell because the surface of the cell is negatively charged. Very commonly, we see things like crystal violet, which would be purple, methylene blue, saffronin, which is a pink or red, depending on who you're talking to. The other type of dye you could use is an acidic dye. The chromophore is an anion, so it's negatively charged. It's repelled by the bacterium. So this is when, I think when you do a negative stain in the lab, you use nigrosin. It's a black dye. And these are, it's not hard to do the procedure, but it can be technically challenging to get it to look right. Too thin of dye, you won't be able to see the slide. Too thick of dye, it'll start to crack and you can't see your cells either way. So to get the right amount of dye so that you can see the cells. So how do you see the cells if you're not staining them? Well, you've stained the background and the cells repel the dye so you can see the little hole where the bacterium is. And again, when done properly, a negative stain is actually, in my opinion, more useful for looking at morphology than a direct stain is, directly dyeing the bacterium. You can really differentiate, especially some of those bacteria that kind of have an intermediate morphology. Is it a coccus or a bacillus? Is it kind of in the middle? You can see it really well with a properly done negative stain. We also could use things like that are called mordants if we want to hold the stain. Have you done a gram stain yet? Okay, so you'll use a mordant when you do a gram stain. It's grams iodine. You also would use a mordant if you wanted to stain flagella. They're very, very thin, so if you use the mordant, it can coat the flagella to make it appear large enough so that you could visualize it under the microscope. So all of these things have a purpose, and we do them in particular order to get a visualization that we can interpret. Have you done simple stains yet? Yes. You're doing them this morning? So afternoon people, you're like, no, we haven't. So half the class is like, no, and half of you are like, yes. And I'm going, oh my God, how many of you are in the lab? Okay, that makes sense. Okay, so you'll do it today, or you've done it this morning. So again, that simple stain. Simple stain means you use one dye. Direct is you stain the bacterium with a basic dye. So remember, the chromophore is going to have a positive charge, be attracted to the negatively charged cell, and you can see probably crystal violet, methylene blue, and saffronin. So it should, again, in theory, if your microscope is clean and you've focused it correctly and you've got a nice amount of light going in, you can see where you have individual cells. These are rod shaped. And then this is a negative stain, and again, it appears that they've used nigrosin because it's black, and so you can see, right, that the cells appear colorless because they are. The dye here is negatively charged. It is repelled by the negatively charged cell. Now, If you're doing simple stains today, you will do differential stains next week. So what you'll be doing next week in the lab, differential stains. You will use at least 2 dyes in order to distinguish different kinds of bacteria, often based on their cell wall properties. And the 2 that are really common for this are the gram stain, which I already told you you have to do for your midterm. Not here, right? You do it for your midterm in the lab. And the acid fast stain. So the gram stain is the most common, and it's the one that you'll hate, and you'll be really mad about it, and that's okay. The more you practice it, the better you'll get. And I know your instructors will give you plenty of opportunity to practice it between now and the lab midterm. This allows us to differentiate whether or not a bacterium is what we call gram positive or gram negative. That distinction, right, gram positive versus gram negative, has to do with the way the cell organizes its wall. In a gram positive cell, you have your cell, it has a membrane, and then right outside of that membrane, it has a very thick layer of peptidoglycan. So remember, the cell wall of bacteria is made of peptidoglycan, thick layer of peptidoglycan. Gram-negative cells, you have a cell, has a membrane, has a very thin layer of peptidoglycan, it has a whole nother membrane on the outside. So gram-negatives are like a sandwich. 2 membranes with a little thin layer of peptidoglycan in the middle. Gram-positive has that thick layer of peptidoglycan on the outside. So it's not just an exercise in frustration. Gram staining and knowing the composition of the cell wall is very important in a clinical setting. Does anybody know why? Penicillin and its derivatives target formation of the cell wall. What we tend to see is that gram positives, right, they just have the membrane and then the cell wall. When they don't have their cell wall, they explode because of osmotic pressure. Gram negatives, they have a membrane, a cell wall, and another membrane. When they don't have their cell wall, they tend to just sit there. They'll bubble up. If they're like a bacillus, they'll turn around because there's nothing giving them shape. They don't explode as much. Yeah, there are some that you can kill them, but not always. So gram-positive cells tend to be more sensitive to penicillin and its derivatives. Gram-negative are not. That might be very important for your patient. The other reason that it's important to know is that in that outer membrane of the gram-negative cells, they have a little toxin. All gram-negative cells have the same toxin in their outer membrane. And it's the one that can often cause septic shock. So if your patient has an infection with a gram-negative bacterium, they're far more likely to develop something like septic shock versus a patient who has an infection with a gram-positive. Not impossible, but far more likely that gram-negative. So it's important as a clinician to understand those differences. The gram stain is the easiest way for us to quickly figure it out. Once you get good at doing a gram stain, it literally does take about, from start to finish, like 10 minutes. It's not that hard. You make your smear. The longest part of that is just waiting for it to air dry. Right? Make your smear. You add your first dye. Right? That's why it's a differential stain. There's at least 2 dyes. First dye, crystal violet. Crystal violet is a basic dye, so it directly binds to the bacterium. It's purple. So right now, if you looked at the microscope, everything is purple. Next, you add the mordant, which is iodine. The iodine binds to the crystal violet and helps it really stick inside of the peptidoglycan. Makes this big crystal violet iodine complex that really gets kind of trapped there in the peptidoglycan. If you looked at them right now, they would be purple. The third step is the decolorizing step. This is the one you're going to hate. Sounds really easy. You just drip a little alcohol on there, and you're done. It's like Goldilocks. Too much alcohol, everybody falls apart, and they all lose the stain. Too little alcohol, you don't wash the stain out of the gram-negatives. Gram-negatives, remember, they only have that really thin layer of peptidoglycan. So the idea is if you put just enough alcohol, the gram-negatives should lose that crystal violet iodine complex. The gram-positives should still be able to hold on to it. So people are like, well, how long do I put the dye on for? I'm like, well, it depends on the thickness of your smear, how many cells are on there. Is the moon in alignment with Jupiter? Like, I don't know. It depends. Every time it's different. You get the feel for like when the color change indicates you should stop. So if this is done properly, when you look, the gram-positives will be purple. The gram-negatives will be colorless. We can't see colorless. Maybe I could draw a little outline here, but you can't see them. So we have to use the counter stain, the second stain, which is saffronin. It is also a basic dye. It is a positively charged chromatophore. It's going to be attracted to the negatively charged cell. And the colorless cells will become pink or red. Those cells that retained the crystal violet still will show purple. So it could look something like this when done properly. You get an A on the midterm if it looks like this. And again, I don't want you to get kind of stuck in this idea that, oh, well, the circle ones are always gram-positive and going to be purple, and the bacillus ones are always gram-negative and going to be pink. Like, nope. There are gram-negative circles. There are gram-positive rods. So what I would always recommend is whenever you are practicing a gram stain, you always use as a control a slide that has a known mixed culture. It doesn't do any good to look only at a gram-positive cell on a gram stain because you can make it be purple no matter how you do it. My little drop of ethanol, I'm done. That's my alcohol. Right? If you know it's supposed to be gram-negative, let me make sure I really put enough alcohol on there to get all that stain out. But if you know it's supposed to be mixed, then you know you're doing the right amount of alcohol. So always use a mixed culture when you're practicing your gram stain. And when you have your midterm, it will be a mixed culture. There will be a mix of gram-positive and gram-negative, and your score depends on, does it look like this? Can I see the difference in the colors? So that's the most common differential stain. But again, another misconception, I think, in microbiology is that a cell is either gram-positive or gram-negative. That's it. Bacteria are only gram-positive or gram-negative. That is not it. There's a whole group of bacteria that don't even have a cell wall, so they're not gram-positive or gram-negative because they don't have a cell wall. But there's another group, or a couple groups of bacteria that do have a cell wall, but you can't do a gram stain on them. So you can't say they're gram-positive or gram-negative. And they don't really have that similar like outer membrane type structure that has the toxin in it. Instead, what they have are a bunch of waxy lipids embedded kind of in the wall and sticking out. So you can try to do a gram stain. You're not going to get an answer. These bacteria are called acid-fast. And the waxy lipids are called mycolic acids, which is why one of the most common genera of bacteria that fits under this category of acid-fast are the Mycobacterium. You may have heard of things like leprosy and tuberculosis. Those are Mycobacterium. Nocardia, you probably haven't heard of, and that's okay. They cause skin and soft tissue infections. But either way, they have that same kind of waxy cell wall. So instead, you have to do the acid-fast staining procedure. There's also a, it's called the Zeal-Nielsen, but acid-fast works. So, same idea in principle, right? A primary stain. In this case, we use carbofuscin, it's red. But because of that thick, waxy layer, you actually have to apply heat when you're doing the stain for like 5 minutes. So you heat it while you're staining. Then you decolorize, and the ones that are not acid fast, they give it up real quick. They're like, yeah, no, we don't want this, it's fine. Because there's no mordant, right? There's nothing like holding it in like that. And those acid fast, that waxy layer, it holds onto that dye really well. So then you counter stain with methylene blue. So anybody that's not acid fast will turn blue. So you would see if you had a mixed culture of acid fast and non-acid fast, all of these little red dots are Mycobacterium tuberculosis. Not terribly common in a clinical setting for a couple of reasons. One, Mycobacterium tuberculosis is biosafety level 3, so we don't culture that in a normal clinical setting. 2, it has a replication time of something like 24 hours. It would take you weeks to grow it on a plate, so you could do a culture like this. There are faster growing Mycobacterium species that you could do this with, and in the lab I think you do Mycobacterium smegmatis. So it's a fast growing Mycobacterium. You can practice the acid fast stain. We also have special stains that we can use to distinguish certain parts of a bacterial cell. For example, the capsule. Capsules are very important in pathogenesis for bacteria. 2 bacteria of the same species, if one strain produces a capsule and the other one doesn't, it's almost a guarantee that the one that produces a capsule will cause a really gnarly infection. The one that doesn't, not so much. The capsule allows them to adhere. It allows them to evade the immune system. It's a very, very important factor for bacteria. But it's this kind of like sticky, gooey carbohydrate mess. And it doesn't really stain. You don't stain the capsule. So why do we call it a capsule stain? Basically, you do a negative stain of the background. So you stain the background. You do a basic stain of the bacteria. So you stain the bacteria, you stain the background, and then you can see the halo of the capsule that surrounds the cell. So if you get good at negative stain, you can always practice trying to do a capsule stain with some of your encapsulated bacteria, like strep pneumoniae. One stain that you will do in the lab is the endospore stain. There are some genera of gram-positive bacteria only that form endospores. These are dormant structures that are highly resistant to basically everything. Autoclaving works to destroy them, but that's about it. Boiling doesn't destroy them, freezing doesn't destroy them, radiation doesn't destroy them, drying out doesn't destroy them. They'll live forever. But again, very few genera of bacteria produce them. The 2 that we might worry about in a clinical setting would be bacillus, like Bacillus anthracis, which causes anthrax. You would worry about Clostridium, like Clostridium botulinum, Clostridium tetani, Clostridium difficile, so C. diff, botulism, tetanus. These bacteria all can form endospores. The staining procedure is not terribly difficult, but remember I told you that endospore is very, very resistant to most things, right? So the way that you do an endospore stain is, well, one, you take an old culture because they're not likely to produce an endospore unless there's like a challenge. And if you're an older culture, there are fewer nutrients, there are a lot of waste product buildup. Forming an endospore is a good evolutionary strategy. So you take an older culture, maybe 48, 72 hours, and you do a primary stain that's called malachite green. So you do that with heat. but you actually do it with heat and steam. So unlike the acid-fast stain, you put a little, you'll put a little piece of paper towel on top of your slide, and we call it flooding. You'll flood it with malachite green and hold it over the flame. And of course, you'll start to see the steam come off, you'll see the dye boiling, and it'll start to evaporate away, and you just have to add more and more dye to basically keep it in that steam bath. Right? That's how an autoclave works. Steam. It's not just heat, it's moist heat. So that allows the dye to at least kind of penetrate the outer surface of the spore, turns them green. You can decolorize with water, and then you counterstain with safranin. So in theory, you would see red vegetative cells and green endospores. Oh, this is a really messy one. So like look ahead, see what day you're doing endospore. Don't wear shoes or clothes that you like. Somebody always gets it like everywhere. The facilities probably hates us because they have to like strip the wax and like re-wax the floor every semester because of the dyes everywhere. This is just a messy one. If you use a nice lab coat, don't use a nice lab coat. Use a disposable one that day. Okay. And finally, this is not a stain that you'll perform in the lab, but it is one that you will have to interpret. You will be given prepared slides, and you'll talk about the flagellum. So the flagellum is a structure that some bacteria have. They use it for motility. And you can do a flagellar stain in order to determine flagellum number and arrangement. So you use a mordant to coat the flagella. They're very, very thin. So you kind of have to build up a layer on them so that you can make them thick enough to see under the microscope. So this is a picture of basically what one set of your slides will look like. They're called spirillum. So it's kind of got a spiral morphology, and then it has at both ends, so just like, you know, amphibians live both in water and on land, we call this amphitricious. It has flagella on both ends. So it has a little clump of flagella at both ends. The other one that you get to look at that's pretty cool is Proteus vulgaris. It's peritricious. It has these little flagella all over the cell. They're everywhere. So it can allow you to visualize flagellar number and arrangement. But again, you don't have to perform it, but you will have to interpret it. So that's microscopy. Questions, comments, concerns? Cool. Okay. So you'll have an exam on Tuesday that will cover chapters 2 and 3 because we finished that. I will publish the chapter 3 homework in a little bit so that you have until Tuesday to work on that. If you end up having questions that you want to go over during office hours, I have office hours again on Monday. 9:30, I think. So, or you can shoot me an email. All right, have a good weekend. See you later.